So welcome back. In this video we are going to discuss about the cell cycle phases, the regulation of cell cycle and the agents which are nothing but a chemotherapy agents which act on this cycle and help in our chemotherapy. So I will try to read each and every line, but some lines can be skipped because it's going to be a long video. So in cell cycle there are actually four phases. Which are these four phases? You have M phase. So before the M phase you have G1 phase in which the cell is settling. So it can enter the G0 phase or it can enter your S phase. Once it enter your S phase, in this S phase you have all your DNA replication, which means you have DNA synthesis, and then it undergoes the G2 phase and it undergoes M phase. In M phase you have mitosis and your cytoplasm gets divided. In mitosis the nucleus gets divided, whereas in cytokinesis cytoplasm gets divided, which gives rise to finally two cells. So this is about all this checkpoint control so when these phases are getting from one phase to another phase, let's say you are going from G1 phase to S phase. So you have some regulatory checkpoints which regulate whether the cell has to go into the next phase or not. These are nothing but your cyclin dependent kinase, cyclin dependent kinase complexes and tumor suppressor genes. So checkpoint control transitions between phases of cell cycle. This process is regulated by cyclin dependent kinase, tumor suppressors. M phase is already discussed. It includes mitosis, which is division of nucleus, and cytokinesis, which means division of cytoplasm. Whereas G1 is nothing but growth phase, where the cell grows before the cell divides. It can be of variable duration based on the position of the cell, where the cell is in the body and the what function it has to play. So cyclin dependent kinase, remove all this. Let's say these are nothing but your kinases. Kinases are enzymes. Let's say if they help in phosphorylation of different tumor suppressor genes, thereby regulating your cell cycle. These kinases are dependent on cyclin. So these are known as cyclin dependent kinases. Cyclins are nothing but your compounds which help these cyclin dependent kinases to get activated. So basically cyclins are nothing but your cell cycle activators. So when you have cyclin get attached to your cyclin dependent kinases, these kinases get activated. So when these kinases get activated, they phosphorylase your RP gene. RP gene is nothing but your retinoblastoma gene. This is a tumor suppressor gene. When your RP gene gets phosphorylated, it gets inactivated. So cyclin and cyclin dependent kinase complexes can then phosphorylate the proteins like retinoblastoma to coordinate the cell cycle progression. So when this get phosphorylated, your RP gene is inactive. Your RP gene is nothing but tumor suppressor gene. When your tumor suppressor gene is inactive, your cell undergoes cell cycle. Okay. So now we will go on to tumor suppressor genes. You have some P53 gene which causes P21 induction and cyclin dependent kinase inhibited. So once this cyclin dependent kinase is inhibited, your retinoblastoma gene is less phosphorylated. So when it's less phosphorylated, which means it is more activated. So once it is get activated, then the cell cycle gets stopped over here. Okay. So if there is any mutations in your P53 gene, let's say, or in a retinoblastoma gene, what will happen is this cell cycle suppression will not be there. This inhibition will not be there, so that the cell proceeds in cycle very rapidly. Okay. So you have some growth factors like insulin and PDGF, EPO, EGF. These bind to tyrosine kinase receptor to transition the cell from G1 phase to S phase, which means they promote the cell cycle to go on. So they are known as growth factors. They help in the genesis of new cells. Let's say, so there are actually three types of cell. So let's say permanent cell. The permanent cell will remain always in G0 phase. G0 phase is nothing but a quiescent phase. These cells are always remaining quiescent phase, so the cell does not replicate. These are nothing but your neurons. See, your brain has some neurons, they don't regenerate. If they need to regenerate, you need a stem cells. Okay, some say skeletal muscles. Let's say you have a patient has lost a limb. He cannot regenerate a limb. You need stem cells to regenerate this, because these all cells are in G0 phase. Then you have cardiac muscles and you have RBCs. See, RBCs cannot replicate. See, one RBC can give rise to another RBC. You have hematopoietic stem cells and bone marrow which gives rise to this all new RBCs. Okay, so this is about permanent cells. Let's say stable cells which are quiescent cells. This entered from G1 to G0 phase. See, G0 phase is this phase. So when they enter the G0 phase, say you need some proliferation. Let's say you have some liver failure, or you have donated the liver to other persons. So then what will happen is your cells to other liver cells will be activated, which enter the G1 phase. So from G1 phase they enter the S phase, G2 phase, M phase, and gives rise to new liver cells. Okay, that's how your proximal convoluted tubule act, your periosteal cells, your lymphocytes are also act. See, okay. Next, labile cells never go G1, which means they are in continuous phase of replication only. Because let's say you have bone marrow, your bone marrow needs to always gives rise to new cells, like say RBC and all. So your bone marrow will always on replication. Your gut epithelium, see, your gut is lined by epithelium. It gets turnover every 90 minutes. We have a phase. Okay, so the epithelium gets eroded and you get new epithelium. You have skin, you have always continuous exposure with the environment. You have hair follicles, you have germ cells to get replicate. See, we will see this in this table. Let's say, first remove all this. Let's say you have P53 gene. What does your P53 gene does is it activates P21. What does this P21 does is it inactivates this complex. Okay, the action of your cyclin complex is to activate cell cycle. See, cyclin helps in rotation of your cell cycle. Once you have cyclin dependent kinases have been inhibited, your RB gene will be activated. So when your RB gene activates, it's like checkpoint. So it stops the cell cycle from G1 to S phase. Okay, and also your P53 gene inhibit apoptosis. One hit inhibits BCL2 and BCLXL, which inhibits Bax caspase activation, which means it indirectly helps in the apoptosis of the cell. Okay, let's say you have a DNA damage or you have cancer cell in which you have the DNA gets mutated. So this P53 gene activation will be lost. Okay, in Leaf-Riemann syndrome you have a complete loss of your P53 gene, and in HPV-E6 also, this is a protein in HPV virus. Okay, this also inactivates your P53. So having discussed your cell cycle and the relevant topic to this is chemotherapy. So we will go and jump into chemotherapy. So the important point over here is the most affected cells are which gets the more replication. See, in chemotherapy you are targeting the cell cycle, and the cell which is always in cell cycle are labile cells, which are nothing but your bone marrow, gut epithelium, skin, hair, germ cells. So these cells will be targeted more easily by your chemotherapy. Permanent cells, let's say you have neurons. Your neurons never enter the cell cycle. So if they never enter the cell cycle, the chemotherapy cannot act on these cells. Okay, so jumping into chemotherapy. Yeah, so the same diagram has been given here. Let's say a cell is in G1 phase. Okay, it has entered the so this all blocks, which is your RB gene, P53 modulation, everything is gone, everything is lost due to mutation, and it has entered the S phase, and it has entered the G2 phase and M phase, and this cycle is going on continuously, which is nothing but you have a rapid proliferation of these cells, which leads to cancer. So to counteract this cell cycle, you need some inhibitors which act on cell cycle and stop this. So let's get on from S phase. In S phase, what's happening is DNA synthesis, and what does DNA synthesis requires? Purines and pyrimidines for its proliferation. So if you give a metabolite that can counteract, that can decrease these purines or pyrimidines, or let's say you have given a purine that can't undergo DNA synthesis. So this is the concept between antimetabolites: scleroderabine, citarabine, 5-fluorouracil, hydroxyurea, methotrexate, pentosin, thiopurines. All these act at the level of purines and pyrimidines, so that your DNA cannot be synthesized. Okay, topoisomerase. These act on both on your S phase and G2 phase. Etoposide, tenoposide, irinotecan, topotecan. We will discuss going on. These are nothing but your antibiotics, anti-cancer antibiotics. So these act on your G2 phase and also your mitosis phase. You have microtubules. What is the function of microtubules is they will help in the division of the replicated DNA so that you have two cells. Okay, see, let's say this is the newly formed your DNA. These microtubules will form here and pull out this DNA. Okay, so that your cell get divided easily. if you stabilize these microtubules, or if you inhibit these microtubules, the cell, it can't pull its own DNA, so that the two cells cannot be formed. And these are cell cycle independent drugs, which means these can act at any phase of cell cycle. These include platinum compounds, alkylating agents, busulfan, nitrogen compounds, nitrous ureas, procarbazine, and actinomycin. Okay. See, this is the same concept. Your antimetabolites act at nucleotide level. See, first you have methotrexate, fluorouracil. These inhibit thymidine synthesis. You have thiopurines. So this is nothing but purine. This inhibit the new synthesis of your purines. And your hydroxyurea inhibit ribonucleotide reductase. Your methotrexate also inhibit the dihydrofolate reductase, so that you don't have tetrahydrofolate. Okay. Your DNA. See, your alkylating and platinum compounds. What these alkylating and platinum compounds does is they just give an alkyl group. They donate an alkyl group. So when they donate an alkyl group, there will be cross-linking inside the DNA. Let's say this is the DNA. So to replicate a DNA, your DNA has to open up. Okay. Let's say you have alkylated it. What will happen? There will be formation of supercoils and cross-links between the DNA, so that your DNA can't replicate. So in bleomycin, your DNA strand will be broken down in anthracyclines and actinomycin. These are nothing but antibiotics, anti-cancer antibiotics. These also break the DNA, and you have etoposide, tenoposide. Remember this by mnemonic, two sides. There are two sides of a coin, so two sides. Likewise, etoposide inhibits your topoisomerase 2. Irinotecan, how to remember this is irino T-con. Okay, you have one topoisomerase 1 inhibitors, and once the cell forms DNA, and then it can form RNA and proteins, or it can enter the cell division. So you have cell division inhibitors, which are nothing but microtubules, inhibits microtubule formation, and which inhibits the microtubule disassembly. So the mnemonic core here is micro-win-tax. Okay, these are nothing but 3 words: micro, win, tax. Okay, yeah. You have antibody drug conjugates. What are these? So once you give a chemotherapeutic agent, let's say you have given a trastuzumab. Okay, this is an antibody against your AHER2 positive breast cancer. You need an agent which can specifically take this to your cancer cells, and which spares the healthy cells, so that you have a definite action at the cell itself, so the healthy cells will be spared. This increases the efficacy and decrease the toxicity. Okay, yeah. Now coming to anti-tumor antibiotics. Simple. So where does your anti-tumor antibiotic acts? It acts at the level of G2 phase and M phase. Okay. So actinomycin is cell type non-specific, bleomycin and erythromycin are G2 or M phase specific. Now so let's say you have bleomycin. How does this act? See, in G2 phase, what has happened is the cell has already formed its DNA in S phase. Okay, there is already formed DNA, which is daughter DNA. You need to kill the DNA. How do you kill the DNA? You induce free radicals formation. If these free radicals are formed, this breaks the formed DNA. Okay. So these are used in testicular cancer, in Hodgkin's lymphoma. This can cause side effects. These side effects are important: pulmonary fibrosis and skin hyperpigmentation. How do you remember this? So you blow the fire. Okay. What is meant by fire? Flagellate pigmentation, ichthyosis, and Raynaud's phenomena. These 3 are important. Remember this mnemonic: blow fire. Okay. So you have dactinomycin. Your dactinomycin also does the same, but your dactinomycin is cell non-specific, which means it can act at any level of your cell cycle. Let's say it acts at the level of your after S phase. Let's say it acts as G2 phase. So it has already formed the DNA, so it intercalates into DNA. So it can act at the level of RNA synthesis. So this is used in Win's tumor, Ewing's sarcoma, rhabdomyosarcoma. It causes myelosuppression. So the similar drug to dactinomycin is mitomycin. Mitomycin is nothing but your antifibrotic drug. So this antifibrotic drug, so let's say you have a stenosis of your larynx, it's nothing but subglottic part of your larynx. It can cause stenosis. So in this case, you give mitomycin. Once you give mitomycin, there is no fibrosis of your larynx. Okay. This can be used as intravesical bladder cancer. Side effects include H2S and TTP. Okay. Now coming to anthracyclines. So anthracyclines also similar action to pleomycin. By the time they are acting, they already have a doctor DNA which is found at S phase. So they generate free radicals in intercalated DNA, decrease the DNA. Okay, breaks the DNA, thereby decreasing replication. They also inhibit topoisomerase 2. Topoisomerase 2. Okay, so they are used in solid tumors, leukemia, lymphoma. So there is a mnemonic over here. You have doxorubicin, daunorubicin. Ruby broke your heart. Will broke your heart when you recall. When you recall, what does this mean? See, heart breaks when you recall Ruby. Heartbreak, nothing but your dilated cardiomyopathy, and you have recall dermatitis. Okay, radiation recall dermatitis. These are two of the specific side effects of your anthracyclines. This is very important. Your dilated cardiomyopathy, doxorubicin and daunorubicin. Now let's say you are in a S phase of your cycle. What's happening in S phase of cycle? There is a DNA replication. So you need to interfere with the DNA replication. How do you interfere? By decreasing the synthesis of purines and pyrimidines. Now these are nothing but your antimetabolites. Let's say you have thiopurine. There is in the name itself they have thiopurine. They inhibit the synthesis of purine. See, they decrease the synthesis of de novo purine synthesis. See, this azathioprine is converted to 6-mercaptopurine, which is an active drug. Okay, this 6-mercaptopurine is activated by HGPRT. And there is another important thing is 6-mercaptopurine. Once the action is completed, it is converted into inactive form by xanthine oxidase. Okay, so 6-HGPRT is for the further action and improving the action of 6-mercaptopurine by forming metabolites, whereas xanthine oxidase degrades your 6-mercaptopurine. Let's say you are using it with yellow purinol. What will happen? Your xanthine oxidase will be inhibited in yellow purinol. So the 6-mercaptopurine is increases and side effects increases. And the another effect of this your thiopurines is they are anti-inflammatory, so they can be used in rheumatoid arthritis, which is an inflammatory condition, inflammatory bowel disease, SLA. These are all autoimmune conditions, right? In ALL and steroid refractory diseases. They are also used for preventive organ reactions. So the main aim of using this is basically your immune suppression rather than your anti-cancers. Okay, you have cladribine and pentosine. They are also purine analogs. See, so they unable to processed by adenosine deaminase interfere with DNA synthesis. Your C is not for C. How do you remember? Cladribine is for HCl, fludarabine is for CLL. C is not for C. And pentostatin, hairy cell leukemia. And see, myelosuppression you need not to remember specifically because everything has myelosuppression. These are the general side effects. Your myelosuppression, your germ cells will decrease. All these are diarrhea, vomiting. All these are non-specific side effects. The specific side effects of 6-mercaptopurine is liver toxicity. This is important. Okay. Yeah. Coming to cytarabine. See, cytarabine is nothing but arabine of phosphorynyl cytidine. This is pyrimidine analog. We have discussed about purine. Now we are jumping into pyrimidines. What are pyrimidines? Cut the pyramid. Cytosine, uracil, thymidine. These terminate the DNA chain termination and inhibit the DNA polymerase. These are used in leukemias and lymphomas. We have a similar drug which is gemcitabine, which is used in pancreatic cancer. How do you remember is let's say this is a pancreas and you have germs in your pancreas. Okay, germs. Yeah. Next you have 5-fluorouracil. See, this 5-fluorouracil also inhibit the pyrimidine synthesis. How does this inhibit? See, if for the formation of thymidine, uracil is present in RNA, whereas thymidine is present in your DNA. For the formation of uracil into thymidine, you have it to methylate. So if you inhibit this enzyme, your uridine cannot be methylated into thymine. Once you don't have thymine, you don't have DNA synthesis. That's the basic concept over here. These are pyrimidine analogs bioactivated to 5-fluoro-DUMPs, which are converted to thymidine synthesis, which leads to thymidine synthase inhibition, which decreases the ATMP and DNA synthesis. Okay, now these are used in colon cancer, pancreatic cancer, actinic keratitis is nothing but your precursor of your squamous cell carcinoma. These are effects are enhanced with the addition of leucovorin. This is important. And you have hand-foot syndrome over here. See, flu. 5-fluorouracil have foot. Okay, 5-fluorouracil hand-foot syndrome. You have hydroxyurea. Hydroxyurea inhibit ribonucleotide reductase. Once you have no ribonucleotide reductase, you have decreased DNA synthesis. They are used in myeloproliferative disorders. Okay. Methotrexate. See, this is important because when you have dihydrofolate, it needs to convert it to tetrahydrofolate, which is converted to 5-10 tetrahydrofolate. This 5-10 tetrahydrofolate helps in formation of your thymidine. Okay. So if this dihydrofolate reductase acts at this level, your methotrexate inhibits this dihydrofolate reductase so that you don't have the formation of tetrahydrofolate and thymidine eventually. So these are used in cancers like leukemias, lymphomas, choriocarcinomas, sarcomas. Okay. You have drug of choice for choriocarcinoma is nothing but your methotrexate. In non-neoplastic conditions like ectopic pregnancies, medical abortions, rheumatoid arthritis, and psoriasis, vasculitis. Okay. Myelosuppression. See, the myelosuppression is occurring because you have no folate, tetrahydrofolate. This is nothing but your like megaloblastic anemia. Once you give leucovorin is nothing but your folic acid. Once you give folic acid, this effects can be reversible, which means all your myelosuppressions will be gone, and you have formation of your RBC, WBC in normal way. You have hepatotoxicity, mucositis, pulmonary fibrosis, folate deficiency, which leads to teratogenic. So these are contraindicated. Actually, these all drugs are contraindicated in pregnancy mostly, and nephrotoxicity. Okay. Now alkylating agents. See, alkylating agents. Remember this by bus, car, nitro. You need nitro engines for your bus and cars. Busulfan. The primary action of alkylating agents, as we have already discussed, they donate an alkyl group to N7 guanine of DNA. Once they give an alkaline group, what will happen is alkyl group, there will be crosslinks in the DNA. The DNA will be forming like this in superstructural coils, supercoils like structures, so that your DNA cannot be replicated. The important point only to remember, it causes Budd-Chiari syndrome. Busulfan causes Budd-Chiari syndrome. Busulfan. Okay, Budd-Chiari syndrome. Budd-Chiari syndrome is nothing but veno-occlusive diseases of your liver. Okay, nitrogen mustards. These are cyclophosphamide, ifosfamide. See, you have a microsomal enzyme which is nothing but CYP3A4 enzyme. This is converted into 4-hydroxy cyclophosphamide, and it forms an acrolein and aldophosphamide. This acrolein which is the agent which causes hemorrhagic cystitis in the patients using cyclophosphamide. So this is the toxic agent. This is the agent which helps in your cross-linking of your DNA. These are used in solid tumors, leukemias, lymphomas, rheumatoid arthritis, rheumatic diseases and all. See, these cause myelosuppression. This is important. Fanconi syndrome. This causes Fanconi syndrome, which is caused by ifosfamide. See, hemorrhagic cystitis. This hemorrhagic cystitis is caused by your acrolein, and also bladder cancer. This both can be prevented by usage of Mesna. Okay, Mesna is for prevention. There is another drug when we go into here. See, when we enter into something called, wait, I'll show you. Okay, I will discuss when we go further. Okay, yeah, in nitrous ureas, you have car Mustang, low Mustang. Your Mustang, you have car, and you need to fill the nitrogen, and you turn over the globe. What does this means? Glioblastoma multiforme. Put nitro in your Mustang and travel the globe, and you travel slowly. So this leads to delayed neutropenia. Okay. Procarbazine, mechanism of unknown. It's used in Hodgkin's lymphoma. So since we have Hodgkin's lymphoma, let's discuss some of the regimens which are used in, which are commonly used. Let's say in Hodgkin's lymphoma, you have ABVD regimen. This includes adriamycin, bleomycin, vincristine, and dacarbazine. Hodgkin's lymphoma counterpart is non-Hodgkin's lymphoma. You have R-CHOP regimen, rituximab, cyclophosphamide, hydroxydonorubicin, and oncovarin. Oncovarin is nothing but leucovorin, which is rescue. Okay, next you have the similar regimen, which is RFC. You have R-CHOP regimen, RFC regimen. See, the R and C are similar, rituximab and cyclophosphamide. The only difference is you have fludarabine and CLL. Okay, now platinum compounds. Platinum compounds also have similar action. They cross-link the DNA. And the important point over here is they are nephrotoxic, and this nephrotoxicity is prevented with amifostine. See, we have prevented the cyclophosphamide side effect of hemorrhagic cystitis with Mesna, whereas here we are preventing it with amifostin. How do you remember? See, mesna has N in it, so these are used in nitrogen mustards. See, N and M, nitrogen mustards. Where have your amifostin doesn't have has P. I remember this is amifostin. P is nothing but platinum compounds. Okay, and this does not help in peripheral neuropathy. This does help in nephrotoxicity, but does not have any action on your peripheral neuropathy. So micronubules. See, once you have entered the M phase, all the cell has all these things have been done. You have formed all your DNA, and you have a cell which is ready to divide. So you are acting at the level. So when you are acting at this level, the only point you can act at is microtubules, which inhibit the division of your cell. So these are nothing but taxanes. They stabilize the polymerase microtubules and prevent mitotic spindle breakdown. These are used in various tumors. They help. The important thing is glove stop deformity. How do you pay tax? You pay tax by hand, and the hand is nothing but glove. Okay, glove stop deformity. Vinca alkaloids, they bind to beta tubule and inhibit its polarization into microtubules and prevent microtubule formations. Okay, these are used in different tumors. The important thing is vincristine crisps your nerves. So when it crisps your nerves, it leads to neurotoxicity, and this neurotoxicity leads to foot drops and SIADH, and it leads to constipation. Since it is causing neurotoxicity, there will be decreased peristalsis, which leads to paralytic ileus. Vinblastine blasts the marrow, which helps in myelosuppression. Topoisomerase inhibitors all causes increase the DNA degradation. All cause increased DNA degradation, resulting in cell cycle arrest in S and G2 phase. Okay, topoisomerase. See, we have discussed TCON, isomerase 1, both sides, topoisomerase 2. These are used in colon cancer, ovarian cancer, testicular cancer, and myelosuppression and alopecia. All we know. The important point what I forgot to mention is this one. The platinum compounds are used in your germ cells cancer, which are testicular cancer, cervical cancer. Okay, which are nothing but your reproductive cancers. This is a drug of choice for your cervical carcinoma, platinum compounds, and also your retinoblastoma. Okay, so having discussed all this, we'll discuss antibodies later on. See, chemotax. How do you counteract this effect? See, you have amifostin. How do I said it? We need to remember this amifostin. So this helps in prevention of your nephrotoxicity. Is a free radical injury, free radical scavenger, and it's helping it from nephrotoxicity from platinum compounds. And you have dexamethasone. These are nothing but iron chelator. Where do we use this? Dexa, daunorubicin. See, the names are similar. Okay, so you have cardiotoxicity from anthracycline, which are nothing but daunorubicin, doxorubicin. We also have recall dermatitis. See, there is a mnemonic over there. I already discussed. Recalling about ruby is harming your heart, like that. See. Yeah, heart breaks when you recall about ruby. Okay. Yeah, you have leucovorin. Leucovorin is nothing but your folinic acid. This is a tetrahydrofolate precursor. This is used in myelosuppression from methotrexate. That myelosuppression from methotrexate is reversible, and it also see in methotrexate we use it for counteracting your side effects. Whereas with 5-fluorouracil, you are using to enhance the effects of 5-fluorouracil. Mesna is also nephrotoxic, which prevents it from nephrotoxicity from cyclophosphomides and hemorrhagic cystitis. Rasburicase is used for tumor lysis syndrome. Okay, on and set. So up to here you have some specific side effects, and these are non-specific, which means any chemotherapeutic agent can cause all these effects. Let's say you have tumor lysis syndrome, it is present in all. For that you have rasburicase. Let's say you have vomitings after chemotherapy, then what we give is ondansetron, granisetron. These are nothing but 5-HT3 antagonists, and prochlorperazine and metoclopramide. These are D2 receptor antagonists. Okay, and you have delayed vomiting, which means after 24 hours. Let's say after 24 hours, then you give NK1 receptor antagonist. These are all antagonists. You need to remember this simply. See, filgrastim. The name itself indicates grass. which means granulocyte stimulation factor. These are nothing but granulocyte stimulating factors. They help in neutropenia. Okay. Apoetin alpha, which is nothing but your erythropoietin alpha, which is used in anemia. And you have in diarrhea, we use palifermin. So coming to the last, so once we can recap all these side effects easily by this tumor man. Okay, so let's say you have Cs. What does this Cs indicates? This indicates ears, cyclophosphamide, cisplatin, carboplatin. Okay, now you have cerebellum over here posteriorly. This includes ceterabine, which is cerebellar toxicity. Okay, now you have vincristin. Vincristin is causing crisps your nerves, which causes peripheral neuropathy. Let's say you have bleomycin and busulfan. These are causing pulmonary fibrosis. Your bleomycin mnemonic is FIR, which is causing flagellate pigmentation and also your Raynaud's phenomena. Doxorubicin, daunorubicin are causing cardiotoxicity. Okay, doxorubicin, daunorubicin, they also cause recall dermatitis, radiation recall dermatitis. Trastuzumab also cause cardiotoxicity. How to remember it? When you touch someone, they break your heart. So trastuzumab cause cardiotoxicity. Cisplatin, carboplatin, same side effects. They also are nephrotoxic. Cyclophosphamide causes CY. Cyclophosphamide causes hemorrhagic cystitis, which can be prevented with MESNA. So these are some others. See, 5-fluorouracil, flow, foot. Tax you pay with hand, gloves, dog. Bleo, blow, fire. Okay, non-specific common toxicity nearly of all cytotoxic chemotherapies include myelosuppression, neutropenia, anemia, thrombocytopenia, GI toxicity, and alopecia. So this is about chemotherapy.